Welcome back, everyone. This is lecture number 22. Today, we're going to talk about emerging viruses. What is an emerging virus? A causative agent of a new or previously unrecognized infection, and also could be a change in a disease pattern. You'll see that one in a moment. And that term, emerging virus, was really coined in the 1990s. I happen to think Steve Morse coined it here at Columbia. Since the rise of agriculture about 11,000 years ago, new infectious agents have gone from animals to people. And that why 11,000 years ago? Because that's when agriculture began. That's when populations of humans could be sustained that were bigger and bigger. Before then, we were hunter-gatherers, 100, 200, 300 people lived together. But now thousands and thousands of people because we could feed them. And that's when viruses started crossing over big time into people. However, it was only in the 90s that we became good at detecting new viruses. And so hence, scientists made up the term, and of course the press loves killer viruses that are going to wipe out humanity. They're really good about writing about those. Another definition for an emerging virus is a virus with an expanded host range and a disease not previously obvious. An example is Zika virus. Zika virus was identified in 1947 in Africa. Yet in 2015, we saw for the first time that it could cause microcephaly. We never saw that before. And so that's an emerging disease. That's why it says here, not previously obvious. I don't think the virus changed. I just think we never saw the microcephaly before. Yet that falls under this category of emerging. Well, most of these new infections occur by transmission of a virus either from a wild or domesticated animal to humans. We call this a zoonosis. When a virus first goes into people from an animal, it's a zoonosis. And sometimes this kind of cross-species infection may establish a new virus in the population of humans, like SIV moving from chimps to humans to form HIV, which we'll talk about next time. Or SARS-CoV-2 is now a human virus. It was once probably a bat virus. Many times, however, cross-species infections are not sustained in people. They do not become human viruses, like Ebolaviruses going from bats to humans, which we'll talk about today, and MERS coronaviruses from camels to humans. These do not become human viruses. They don't transmit from human to human very effectively. Now, one of the first descriptions of an emerging virus was in the 60s, this book called Fever by John Fuller. This described the emergence of Lassa virus in Nigeria. And this is a book that got me to be a virologist. I know I've told you that before, but I just was looking for something to do as a recently graduated biology major. I was working in a lab. I read this book, and that was it. You have this book to thank me for teaching you today, so you might want to read it. It's really good. A lot of it takes place at Columbia. Here we go. Ancestral origins of human pathogens. Human viruses from 31 genera have crossed over from animal sources and have become human viruses. That's the orange bar here. So they transmit efficiently among humans. Viruses from 37 different genera are what we call zoonotic pathogens with little or no ability to transmit among humans. That would include Ebolavirus and others that we'll talk about today. The remaining pathogens have co-speciated with humans. Viruses from 16 genera were vertically transmitted from ancestral hominin species at the emergence of the Homo genus. In other words, we got it from our ancestors, and they probably got it from some animal. And viruses from 6 genera were vertically transmitted from related Homo species, like Neanderthals, to us. So the 16 are non-Homo species, and the 6 are Homo species. Even these 16 plus 6, they came to the Homo ancestors or the hominin ancestors from another animal. As I said last time, every human virus came from an animal at some point. Why is this? Why do viruses cross over from animals to humans? Well, here are the overriding factors. Amazing population growth, and because of that, change occurring in all the ecosystems that we do. We screw it up. We mess up the planet. We invade animal habitats, and we get their viruses. I mean, look at the population growth. More people need more protein. They need animals, and they invade other parts of the planet, as you'll see today. Here's some of the activities that promote virus emergence. A lot of people, of course, but also, I mean, look at this travel. This is I took this screenshot at some website where you could see all the planes in the air at any one time. This was done in 2019. And obviously, air travel brings viruses everywhere, but also other things you might not think of. You know, dams, water impoundments, irrigation, deforestation, rerouting of wildlife migration, wildlife parks, long-distance transport of livestock and birds. Amazing how much viruses they carry. Of course, blood transfusions, xenotransplantation, putting animal parts into humans, societal changes with regard to drug abuse and sex, huge increase in drug abuse and so forth. Urbanization, daycare centers is a big one. Hot tubs not properly sanitized, air conditioning because of the water used for chilling, and then, of course, the used tires, which I've told you about, transport mosquitoes. And don't forget climate change, which is raising the temperature of the globe and natural disasters. So viruses are everywhere, as you know. I said it on day one, and they're there for us to encounter. And this is just one slide to emphasize this. The Amazon North region of Brazil, which you know is being deforested, so people are going in and working. You know what? There are animals that live there, lots of animals, and these little words, which you can't read and you don't have to, each word is the name of a virus that's been discovered in this region. So we have been able to isolate viruses from all these regions. It's like 183 different arthropod-borne viruses that live, that reproduce in animals in the forest. And that's just the ones we know about. I can tell you there are far more. That's why they do things like this. These people are going to be infected with viruses that they encounter during the deforestation. We know for a number of viruses some of the factors that have led to their emergence, and this is a table that shows some of them. Dengue virus we almost wiped out, but it came back because of the tire trade. Ebolavirus, the bushmeat trade in Africa, we'll talk about them today. Hantaviruses, human-rodent contact during rice harvest in many Asian countries. Hendra virus, proximity of fruit bats to horses and stable workers. HIV, hunting and butchering of infected primates. Bushmeat trade again in Africa. Influenza virus, pig and bird farming. And then we'll talk about today MERS, Nipah, SARS, SARS-2. We will talk about West Nile, mosquito transmission from bird reservoir, global travel. Remember, it wasn't in the Western Hemisphere until 1999. It came from Israel to the U.S. And Zika virus. So these are the factors on the third column. And for SARS-CoV-2, we don't yet know what the factors are, but we're going to try and find out, of course. Now, don't forget that evolution is incredibly important for emergence. You have to think of every animal out there that's full of viruses. And you know, for most of the spillovers we're going to talk about, we're talking about a bat, a pig, some kind of a bird, non-human primates, rodents, camels, you know, animals that are close to us, not insects, not snakes. Probably the viruses that infect them are too distant. But in each of these hosts, there's a huge biodiversity of pathogens. There's a quasi-species. Remember, we talked about that last time. In every animal, the viruses exist at quasi-species. That means they're randomly mutating, and randomly mutations may emerge that allow that virus to infect a new host. And that's what I mean by adaptation to new hosts and environments through variation and selection. So what's reproducing in a chimp could infect a human. Of course, it has to encounter the human. That's part two. So you have quasi-species and encounter. Otherwise, you're never going to get anywhere. Let's talk about different categories of interactions between hosts and viruses. And remember, you know, you think about all these viruses and someone's asking, how likely are they going to be able to cause severe disease? Who knows? You want to take the chance and have a pandemic like we have now? Obviously, most of them don't go anywhere. But now and then, the right one is going to get into people. It's going to spread a little bit in people and then get better adapted. We'll talk about that in a moment. First, I want to talk about 4 different categories of host pathogen interactions. We have them shown in this picture, which you're going to see for a few slides. Stable, maintains the virus in the ecosystem. Stable virus-host interaction. Then we have evolving. This is what happens when a virus is passed to a naive population. It could be the same host that's never seen the virus before, geographically diverged or different place, or it could be a different host. And then the virus and the hosts evolve for a while, and then they become a stable relationship, perhaps. We have dead ends, one-way passage to different species. Maybe it will reproduce a little bit, and that's the end of it. Or the resistant host, the infection's blocked, never gets anywhere. The virus just does not reproduce at all. So let's take a more in-depth look at some of these. So stable, here in green. So the virus and the host are both existing. They multiply. The virus multiplies, the host multiplies. We have these arms conflicts that we talked about last time that maintain both in a stable fashion. And some of these are permanent. Like humans are the sole natural host for measles virus, herpes simplex, cytomegalovirus, smallpox, and many others. So we are in a stable relationship with those viruses. And sometimes there's more than one species involved, not just humans. Influenza A viruses, humans and other species. Flaviviruses like Zika virus in West Nile and birds, and dengue virus and other arthropod-borne togoviruses. Now next, the evolving host relationship. Unstable and unpredictable. Those are the hallmarks. It's hard to know what's going to happen. And the outcome of infection can be nothing or it can be death. Some examples, we'll talk about this one, the introduction of smallpox and measles to natives of the Americas by Old World colonists and slave traders. Remember, for a long time, the New World was isolated. Hardly anyone went there. It was really hard to get there. Boats were not very common, no airplanes. But then when people started exploring, then they brought their viruses with them and an evolving host-virus relationship. Introduction of West Nile into the Western Hemisphere. When it got here, it spread like crazy across the U.S. and up and down North America and South America. The interaction was unstable for a while and then stabilized. And, of course, the introduction of myxoma into Australia to eliminate rabbits, first year, wiped out most of the rabbits. After that, it started to stabilize. That's what I mean by an evolving host-virus relationship. It's a transient period after when the virus is introduced into a new population. Dead-end interaction. This is a little confusing every year for most students. Frequent outcome of cross-species infection. We'll see some examples of that today. There's no sustained transmission from the new host to others of the same species. Ebolavirus probably has a reservoir in bats, mainly in Africa, and it may accidentally infect humans, chimps, and gorillas. These are dead-end infections. However, the West African outbreak of Ebolavirus in 2015, over 25,000 cases. It didn't sustain. It eventually stopped, and we'll talk about that a little bit. Influenza H5N1 crosses over from birds into people, never really establishes sustained human transmission. So these crossovers, these dead-end infections, contribute little to the natural history of the infection in nature. The reservoir is where things happen. We call it dead end because there's no sustained transmission, but there is some occasionally, as you will see. Okay, and then, of course, resistant we're not going to talk about because nothing happens. We've already talked about you eating insect viruses with your cabbage. Nothing happens. It just passes through you. Nothing to say. Move on. Let's look at some examples of stable and dead-end host-virus relationships. Here is an example of both with one virus. So we have a virus that is transmitted among wild birds by mosquitoes. Yes, mosquitoes bite birds, not just humans. In fact, mosquitoes bite a lot of animals in the forest. Many mosquitoes bite plants. In fact, the males just drink sap. They don't need blood meals. Anyway, this mosquito bites a bird, it picks up a blood meal, and spreads the virus to another bird. Sometimes Domestic chickens can be bitten and they can receive the virus. Sometimes the mosquito will bite other birds and other mammals. Other mosquito species come in sometimes. It can be a really complex host-virus interaction, but this is the stable part. This is the natural reservoir of this virus. Now, occasionally, as you know, mosquitoes can bite us or they can bite horses, but it's an accident. It doesn't contribute to transmission. These are dead-end hosts. The horses, the humans may or may not get sick, but they do not serve to transmit the infection. For example, West Nile, so the mosquito, if it carries West Nile, is transmitting it among birds. If it infects a horse or a human, they may get ill, but they're not going to have enough viremia so that if another mosquito bit them, they would transmit it. So it's a dead end. Similar situation with many viruses. Here's another example where we have a tick-borne virus. This is a tick that is feeding on rodents in the forest to get blood, and that rodent is infected. The tick picks up the virus and spreads it to another rodent. Over the winter, when there aren't many rodents to bite, the ticks can harbor the virus in the ova, and in the spring can then pass that to the offspring. Occasionally, ticks will bite dead-end hosts like cows. It's an accident. The tick wanders into the field or a barn, it bites the cow, and it's not going to transmit it on to another tick. And if we drink the cow milk, in some cases, we can get that virus, but we're a dead-end host. We don't transmit it. Or the tick can bite humans as well. You're not going to transmit it by another tick bite. All right, dead-end hosts here, these are the natural reservoirs for these viruses. Stable hosts, dead-end hosts. Back to emergence. 2 steps. First, we have the quasi-species in these animals, which we've talked about. That's essential. The fact that just randomly a virus can emerge that might be able to infect another host should it encounter that host. That's the key. So we have to have encounter. If a bat in the forest spawns a virus that can infect humans by virtue of binding to a receptor, it doesn't matter unless that bat encounters a human. So you have to have an encounter. So any of these species with their viral quasi-species have to encounter humans. And as I said, we make it easy. We make it easy to encounter animals, more and more so over the years. And then establishment and dissemination. If a single hunter is bitten or somehow gets infected and doesn't see anyone for 6 months, that's the end. That's the end of the infection, whether or not the hunter gets sick. If someone is harvesting bat guano in a cave and gets infected and never sees anyone else, that's it. However, if that farmer harvesting guano goes home to his family and infects the family, now the virus may start to pass among humans. Maybe it gets selection for better human passage, and then maybe if one of the kids in the family goes into town, then the kid will spread it to more people. You get the picture. You need to have establishment and dissemination. One host isn't enough. Rare chance encounters of viruses with new hosts may never be detected. Maybe they don't work so well. Maybe the host never encounters anyone else. Even if a farmer goes into town after being infected, he may not be making enough virus to transmit it. So these single host infections may not be transmitted among humans for many, many reasons. We'll talk next time about how SIV spilled over from chimpanzees to humans in the 1920s. It probably spilled over many times before that and has continued to spill over. But most of the spillovers don't go anywhere. So it's a rare event that a spillover establishes a new infection in humans. So obviously when a human encounters an infected animal, the virus has to have access to susceptible and permissive cells, right? If the guano gets on the hand of the farmer, the outer skin layers are dead. But if the farmer wipes his or her nose, he could introduce the virus into her nose, and it might reproduce. And obviously, population density is important. As I said, if it's just one person, it's never going anywhere. Families or small towns, etc., make a difference. In health, perhaps the farmer is not so healthy and allows a more extensive replication. But eventually, the only way that spillover establishes a new human infection is by serial infections, by establishing chains of transmission from that initial infected host to more and more and more. And that's what happened with SARS-CoV-2, of course. Somewhere there was an initial spillover and possibly small numbers of infections before we noticed 40 cases of atypical pneumonia in December 2019. And we don't know those early days, very hard to pick up. Let's take a look at some examples now to really amplify these concepts. So we'll start with some very old ones and then move into modern ones. First, smallpox virus. By sequencing lots of human isolates and animal isolates and doing phylogenetic analysis, it looks like smallpox emerged about 3,000 to 4,000 years ago in East Africa, right around there. And we think it came from camels. Now, camels were introduced to Africa 3,500 to 4,500 years ago. They originated in what is now United States, that black dot there, 45 million years ago. And then they went south into the South America, and then they crossed over the land bridge here, and then eventually made their way to Africa. The camel's changing its geographical range. It's going to get infected with new viruses. So what we think happened is, again, looking at the phylogenetics of this, we think camels were infected with an ancestor of smallpox from a gerbil. This is a gerbil here. They had the ancestor of smallpox. When the camels came, it disrupted things. A new host, the gerbil virus, infected camels. And then people started using camels for their agriculture. And boom, smallpox infects people. And then it spread among people, became a human virus, a very serious one. Measles virus. Measles virus, remember? Paramyxovirus, negative strand RNA virus, is closely related to a virus of cows called rinderpest virus. We think we got measles virus from cows, basically, from an ancestral rinderpest when we first began to domesticate cattle in the 11th to 12th centuries. Why? This is when people are starting to learn how to plant crops and how to take certain animals and use them to do work for you. Cows are wild, and someone got the idea that let's take these animals and use them to pull our carts or whatever, and they started to breed them. And of course, they have a barn full of cows, and now in the morning you go to get your cow and you get infected with the cow virus. That's where measles probably originated. It was established in the Middle East when human populations began to grow and congregate in cities. Measles requires populations of between 250,000 and 500,000 people to sustain it. And then once it established here in the Middle East, it spread around the world by people moving elsewhere, colonizing other places and migrating, and it reached the Americas in the 16th century and essentially destroyed many Native American populations because they had never seen it before. They hadn't been doing anything with the cows. Now, in connection with smallpox and measles, we know through history the effects of virus infections on native populations. These are what we call diseases of exploration and colonization. And again, the idea is that the Western Hemisphere was pretty isolated for a long time. They had their own viruses, of course, because they had animal species. They acquired viruses from them, but they grew to adapt to them. Same thing in Europe. As measles and smallpox spread, people became immune, had less severe disease. Of course, when Europeans started to move west and colonize and explore, they brought their viruses with them. And what happens when you put a new virus in a naive population? You have explosive growth. So this is already a human virus, right? From Europe and the Middle East. But you bring it to a population with no immunity, you know what's going to happen. You saw it in the past year. Explosive spread. So smallpox made it to Europe after emerging in the Far East from the camels via the gerbils. And it was horrible disease in Europe. Many people infected, huge epidemics. And then it was brought to the New World. It spread from Hispaniola in 1520, brought there by colonists, and killed 3.5 million Aztecs in 2 years. And that's why Cortez was able to conquer the Aztec Empire, with his miserable band of soldiers because they were all sick. The Aztecs, that is. So it changed the balance of human populations. Same thing with measles. Measles is thought to have been brought over and killed many Native Americans as well. Then we have some interesting stories of emergence. This is my favorite by far because it involves poliovirus, my favorite virus. I call this changes in human populations and environments. Epidemic poliomyelitis began at the beginning of the 1900s. This is a epigraph of cases of polio, annual reported cases, and you can see before 1900, really no outbreaks. But you know, this Egyptian carving from thousands of years ago suggests that it was around then. It seemed to be sporadic. There was a case here and the case there, and you could read it in the medical literature, but never any outbreaks. All of a sudden, 1900, boom, we have thousands and thousands of cases peaking in the 50s with 30, 40,000 paralytic cases a year in the U.S. What changed? So that's an emerging infection because the virus has been around. We know about it. It's a human virus, but yet something changed so that the disease pattern changes. Now we have epidemics. Well, here's the story. Polio is a disease of modern sanitation. Before 1900, the virus circulated freely. Most babies infected shortly after birth because sanitation was horrible, as I've mentioned before. Sewage and feces everywhere. No sewers, no toilets, but the kids were protected by their mother's antibodies. Mother had been infected. Her antibodies protect the child. However, those antibodies don't last forever. But most kids are infected very early on. The antibodies prevent paralysis. We then develop the toilet and sewage, and we improve sanitation. So you're no longer infected at birth. You get infected later in life when your mother's antibodies are gone. And that's where we get epidemic polio. You now have thousands and thousands of kids who are susceptible. You introduce the virus, boom, you have an epidemic situation. That's the reason why polio became epidemic in 1900. But I love to say, had we had genome sequencing in 1908, we would have blamed it on mutation. You can bet how many scientists would say, oh, the genome mutated, and that's why it's causing outbreaks now. Now, I'm sounding a little sarcastic because it's an easy way out to blame the genome. Sometimes it's not so easy. You have to think a little harder. And this was a wonderful story about how, in the absence of having any genome sequencing, we figured it out. And I can tell you that most changes that we see today are likely due to epidemiological factors, not just the genome. Now, another really interesting story involves changing climate and animal populations. This is a disease called hantavirus pulmonary syndrome. A related disease had been seen for many years in Asia, but not in the U.S. And suddenly in the Four Corners area of New Mexico in 1993, so right there where the four states come together, there were suddenly cases of severe pneumonia, respiratory disease, and otherwise very healthy young people. And many of them died. So it was called eventually called hantavirus pulmonary syndrome. And the virus was identified. The name ended up being Cynombre virus. The CDC identified it as a virus of the deer mouse, Paramiscus maniculatus. Now, they originally wanted to call it Muerto Canyon virus because that's the little town, not the town, but Muerto Canyon was right near some of the first cases, but the residents objected. They didn't want a virus named after their region. So they went through a few names and they rejected them all. So they came up with Cynombre, which I think is funny, right? Okay, we'll call it nothing. We'll call it no-name virus. Some places don't like to have a virus named after them. Other places could care less, right? Or other places have no choice. All right, so if you catch Paramiscus, which are all over the U.S., about 30% of them will actually have virus. So you should be careful with wild mice. Okay, so hantavirus is a Bunya virus with 3 RNA segments, envelope virus. So what's the story here? Why did we suddenly see this in 1993? Well, it turns out the previous year there was a lot of rain, and it caused an increase in the amount of piñon nuts, which are delicious, right? I love piñons, and people love them, and so do deer mice. And so the mice had a lot of piñons to eat, so their population increased, and they went into people's houses, right? They just more mice, more places to go. Contact with humans increased. The virus is excreted in feces of the mice. They go in your house, they poop, and dries up, and then you go to sweep it, you aerosolize it, and you inhale it, contaminated blankets or dust, and that's how we inhale it. We didn't have many mice in the house before because there wasn't such a big population, but the increase in rainfall did that. So we're not the natural host. We are pretty much a dead-end host here, not really transmitted among humans. And human disease is pretty rare, although now we recognize it over and over. And we go back into the records, earliest cases, 1959. I think it's ironic that, if you live in Muerto Canyon, right, you don't want to have a virus named after you. Who can explain human behavior? Go figure. So you see here the mice are excreting it in feces. You go to sweep up a mouse poop, right, and you inhale it. So the moral is, if you have mouse poop in your house, don't just sweep it up. First, make it wet. Spray it with some water, or better yet, bleach, right? And then you can sweep it up, but you don't want to aerosolize it. So now the CDC tracks hantavirus pulmonary syndrome. And there have been 728 cases as of January 2017. And this is the epi curve per year. Died. These are people that died in blue. Lived is in green, and the others we don't know. So you can see it goes up and down. It depends on the mouse population. Many cases, not many, but sometimes cases occur when people go camping. And, up in upstate New York and also in California, there have been some cases where, they have these wooden platforms where you could pitch your tent, and the mice live under it. So you pitch your tent, you're inhaling mice feces. The mice that are involved are not just the deer mouse, that's Paramyscus maniculatus, but the white-footed mouse, the rice rat, and the cotton rat. This is the range of Paramyscus. So not a lot of Paramyscus around here, but other kinds of mice can harbor this as well. So you basically need to be careful about mice in your home, and when you go camping. I don't like those platforms. I'd stay away from them. Just put your tent on the ground. All right, let's move to bats. Around the 90s, we learned that we can get viruses from bats. We didn't know that before. And this is one kind of bat that has a lot of paramyxoviruses. It's called a flying fox. It's got a 3 or 4 foot wingspan. It is a fruit-eating bat, so bats can eat fruits, frugivorous bats, or they can be insectivorous bats. They can eat insects. And these guys are frugivorous. They eat fruit. And we'll talk about Nipah and Hendra viruses, which were found in flying foxes in the 90s. And these viruses have been shown to cause severe disease in animals and humans. This is just a paper I wanted to show you, published in 2012, where they did a study of bats and found 66 new paramyxoviruses from 119 bat and rodent species. Paramyxoviruses alone, which is what Nipah and Hendra are. This kind of raised our awareness of the importance of bats in zoonotic spillovers. And of course, since then, it's gotten even more intense, I think. So Hendra virus, first discovered in the town of the same name. So Hendra didn't care. This is Hendra by Brisbane down here. September 94, there was an outbreak at a horse farm, racing horses. And the 14 horses died. This is a horrible thing, right? If you race horses, 14 race horses, it's millions of dollars worth of animals. And the trainer, one of the trainers who took care of them, died as well. When you take care of horses, you're right up next to them. You're inhaling their respiratory secretions. And so that's what happened here. Now, these stables are outside, of course. And what happened, they finally traced this virus to flying foxes. The flying foxes come onto the stables. They fly in at night and they contaminate the horses. They shed the virus in their urine. So what happened here is that the flying foxes infected the horses, and then the horses infected the humans. And this continues to happen. It's not very common. It's quite rare in people, but horses over the years have continued to acquire this infection because the bats can have free access to the stables. So what they have done is to develop a vaccine for horses, not for humans, because the human disease is too rare. You don't know who to immunize, so you just immunize the horses, and that protects humans. This is called a one world health approach. One world means we're all connected. All the animals on the earth Whether you like it or not, we're connected. And if you, in this case, prevent infection of horses, you're going to protect people. That's a One Health approach. This, by the way, is the range of Pteropus, which is the genus of flying foxes here. Very wide range, as you can see, and not just Australia, but much of Southeast Asia and India. All right, so that's Hendra virus. One of the earliest suggestions that bats have viruses that can spill over to people. Then we had Nipah virus, the next one, an outbreak in Malaysia in 1998. This happened in the Perak district of Malaysia, which is right up that red box there, where they had pig farms. The pig farms were outside. This was an outbreak of respiratory and neurological disease on pig farms. The pigs got sick, and then a lot of humans got sick, and 105 deaths. So, the handlers of the pigs got the disease. And this outbreak was stopped in Perak by killing basically 1 million pigs. And it's really unfortunate because it's a big source of protein, of course. And that stopped the outbreak here. And they since moved the pigs indoors, and that stopped them from getting infected. But from the same outbreak, some pigs were shipped down south, and there was an outbreak in this southern area here. And eventually some of those pigs made it to Singapore, and they had some outbreaks in Singapore as well. So stopping the movement of pigs and putting them indoors stopped all that. So what happened here? Again, this virus came from fruit bats, which excrete this virus in their urine. They infected the pigs in the outdoor piggeries, as they're called. They're orchards. The pigs are in orchards with fruit trees, and the bats come to eat the fruit, and they contaminate it, then it drops, and the pigs eat it, and they get infected. So bats infect pigs, pigs spread it to humans. And subsequently, in other outbreaks in India and Bangladesh, it's acquired by consumption of date palm sap. So in these areas, people like to drink the sap from palm trees, and they put these collecting buckets in, and the bats come at night, and they like the sap too. And while they're drinking, they're peeing in it, and they're peeing in the virus, and then you drink the sap and you get infected. Low-tech solution was to cover these traps at night. By the way, there's a great exhibit at the Smithsonian called Outbreak, where they have this. They have a palm tree and a collection, so you can read all about this. It's really cool. There's a little bit of human-to-human transmission, mainly because in many of these areas, people bury their own dead. They prepare the body, and they're very close to it, and they get infected. And these infections now continue in India and Bangladesh, and a vaccine has actually been developed. It's the Hendra vaccine, because the Hendra glycoprotein, which is the vaccine developed for horses, also protects against Nipah. So that has gone through a phase 1, because there are a lot of infections every year, and they do need a vaccine. So why did this happen in the first place? This is an interesting study in ecology. So the reservoir, of course, is the fruit bat of Nipah virus. In the 70s through the 90s, they were deforesting. They wanted pulpwood, and they wanted to clear land for industrial plantations, and this is the habitat for the bat. So the bats get forced out of their habitat. They burn these fields, these forests, and so the haze from this burning blanketed much of Southeast Asia in the few months before this Nipah outbreak. Made worse by the drought driven by the El Nino in that year. Basically, fewer flowers and fruiting trees for the flying foxes. They have nowhere to eat, so they go and they look for food elsewhere. So they go into the cultivated fruit orchards, which is where the pigs were kept. So we basically made them change their habitat by all of this. And having these piggeries and fruit orchards wasn't a good idea. As I said, we've changed that. So here's the web of emergence. This is a really well-documented story. So we have a drought caused by El Nino. We have haze from anthropogenic forest fires as a consequence of deforestation. We remove their habitat. The haze also pushes them out. The bats don't like the haze. Their food supply goes down. They migrate. So we put pig farms in orchard. They go to the orchards and they eat the fruit. They contaminate the pigs, and it eventually gets to us. If we behaved, we could stop a lot of this. I think that's part of the story here. Here's in a nice cartoon fashion, the ecology of Nipah spillovers. So the bats are foraging on fruit trees on the pig farms, and they contaminate the fruit. Pigs get infected. People get infected. That was the first outbreak in Malaysia. And then more recently, the bats contaminating date palm sap, and people drink it and they die. And then people get infected through contact with patients, so preparing the body and these patients can get infected. But then there are some other cases which have no connection with date palm sap. And maybe it's a flyover event, maybe a horse is involved, we're not really sure. So anyway, that's Nipah virus with clear patterns of emergence. One more I want to tell you about, because I think it's another great story. This is Argentine hemorrhagic fever caused by another bunyovirus, Junin virus. The Argentine Pampa, right? A very nice area. They wanted to convert it to farming, to grow maize. So this is like post-World War II. So they used herbicides to control the weeds so they could plant their maize. And by killing all the weeds, they allowed a new shade-tolerant grass to grow in the shadow of the maize. So this grass wasn't there before, but you got rid of its competitors by the herbicides, and now it grows. And guess what? There's a new mouse that comes in to eat this grass, which wasn't there before, Calomus musculinus, and this mouse has a virus that wasn't there before, Junin virus. And so in 1953, we start to see cases of a new disease called Argentine hemorrhagic fever. The virus isolated a few years later. It's all because of what we did to convert this land to grow maize. It introduced a mouse. So this is a story that goes over and over and over again. Our anthropogenic activities lead to new encounters with viruses. It's our fault, folks. Don't blame it on a lab. Ebolavirus. The first Ebolavirus outbreak was back in the 70s, 1976, in the DRC. Each of these outbreaks, and this is a summary of the outbreaks up until the big one in 2015 in West Africa. Previously, they were mainly here in Central Africa. And they were pretty small, as you can see by the numbers. A few cases here and there, a few hundred. And this thing was over 25,000 cases. And there have been a few more recently. There's one ongoing actually in Guinea and in DRC. So what's going on here? Each outbreak actually is a new zoonotic spillover, except maybe for the new one in Guinea, which may have been from a person who is persistently infected. We talked about that before. Each one is a new zoonotic spillover. So Ebolavirus is a negative strand enveloped RNA virus with a pattern of making subgenomic mRNAs, and it has an unusual filamentous appearance. That's why we call them filoviruses. It's a classic zoonosis. And there's usually an index case. Sometimes you can pinpoint contact with an animal carcass of some kind, bushmeat trade. People, you can't go in a supermarket and buy your food. You have to hunt it. So you go into the woods and see what you can get. It's called bushmeat. In some of these cases, you can find contact with a carcass, but not always. Often they just start and you don't know why. So you have an index case and then typically transmitted to others by close contact, either in healthcare situations or, again, burying the corpse of your family. You prepare it and you get infected. Very short chains of human infection. These transmissions are not sustained. That's why we call this a dead end with an R naught of 2. So here are two examples of Ebola virus outbreaks. This first one in Gabon in 1996. There are a number of different kinds of Ebola viruses. This is the Zaire Ebola virus. 37 cases. A chimp found dead in the forest was eaten by people hunting for food. 18 people involved in butchering the animal became ill. 10 others occurred in their family members. That's typical, and we don't get virus. Nobody goes back and looks at virus, so we really can't prove it, but looks like this chimp was the source. Another one the next year in Gabon. Index case was a hunter who lived in a forest. A dead chimp found in the forest at the time was infected with Ebola virus. Well, we don't know if this hunter contacted it, but this is the pattern here. We think it's from bushmeat trade. Here's the early contact tracing on the outbreak in Guinea in 2013. It all began in this small village where there were 9 deaths, and it began with a 2-year-old child here. That's the index case. Fever, black stool, vomiting, died 4 days after onset of this disease. We have no idea how the little girl got it. No idea, but somehow contact with bushmeat. And then look at the chain here, how it works. So that's S1. Her sister died. She's living with her. The mother of the 2 girls died. The grandmother died. The nurse taking care of them died. The village midwife died. And that midwife went to another village and spread the virus there, and another village here. And she also went down here. I mean, you can see the point. And then it goes on and on and on. These chains of infection. So healthcare and family, that's how this infection is spread. Where does it come from? We don't really know for sure, but Marburg virus, which is in the same family, Filovirus, has been isolated from the cave-dwelling fruit bat, Rosetus egyptiacus. Zaire Ebolavirus RNA antibodies have been found in 3 tree-roosting bats. 3! After all the years of searching, but no infectious virus. So we suspect it's a bat, but we probably will never prove it. The bat is most likely the natural host, and humans, gorillas, and chimps are dead-end hosts. They all can be infected. So the origin, bats probably are reservoirs. They periodically infect chimps, gorillas, maybe humans directly from bats. Some people eat bats there. Sometimes humans, look for bush meat in these other species, and they get infected. But these are dead-end infections and very little transmission, very little sustained transmission. Eventually you can stop it. It doesn't become a human virus. The transmission is by contact with blood or body fluids from someone who is sick or has died, contact with contaminated objects like needles and syringes, not by insects, not by water, not by food, not by aerosol. This requires close contact. If you're doing a medical procedure on a patient, you can get it from that. So it will enter mucosal surfaces, breaks in the skins, parenteral via needles. It will reproduce in the skin. You can find it in body fluids, nasal secretions, blood, semen, really reproducing in a lot of different tissues, and an incubation period of 2 to 21 days, during which you're not contagious. In the 2015 outbreak in West Africa, a number of those people flew elsewhere, and they didn't know they were infected because they had no symptoms during the incubation period, and they were not contagious. And it wasn't until, say, some of them arrived in the U.S., then they had symptoms. That's the point of contagiousness, and there were a few transmissions in a Dallas hospital, for example. As I said, this virus involves many systems: gastrointestinal, respiratory, vascular, neurological. It's amazing, and very high case fatality ratio in Africa. It's a zoonosis. It is not sustained. All right, SARS-1. I don't know if any of you look at ProMedMail, but it's a very cool website that tracks outbreaks of all kinds, and you can go to the website and check it every day, or you can have the outbreak news emailed to you. But this is a post from 2003, February 10th, from an MD PhD who was working for this consulting firm here in the U.S. And he writes, "This morning I received this email. I searched your archives, found nothing. Does anyone know anything about this?" So he's writing to ProMedMail, and this is the email he got. "Have you heard of an epidemic in Guangzhou? An acquaintance of mine from a teacher's chat room lives there and reports that the hospitals there have been closed and people are dying." That was the first indication of SARS-1. This is February. First time anyone had heard of it outside of China. It actually started in November, but China kept it pretty close to their vest. An outbreak of severe atypical pneumonia, unknown etiology in Guangdong. 300 cases, 5 deaths. This is November 22. And the characteristics of the disease: 2 to 10 day incubation period, fever, chills, headache, malaise, myalgia, all right, flu-like symptoms, and then dry cough, shortness of breath, and then 10 to 20% of the patients may require mechanical ventilation. Sound familiar? Acute respiratory disease, acute lung injury. The Chinese doctor who had treated some of the first patients in Guangzhou, he went to Hong Kong on February 21st the next year. And he went to this now famous or infamous Metropole Hotel. He died the next day in the hospital. So he had acquired SARS, taking care of patients in China. And he started a super spreader event. The infection spread to 10 other people in this hotel who flew off to different countries: Singapore, Vietnam, Canada, and the U.S. before they had symptoms, and they developed symptoms there and spread it to other people. Eventually, this virus spread to 8,000 people in 29 countries, about 10% mortality, but we were able to stop it then. This is a travel poster from that era. Hong Kong will take your breath away. It's the epi curve. Remember, it starts in China in November. But then around February, they informed people internationally that, yes, they have this issue because of that ProMed mail post, and ended in July 2003. Here are the numbers. Only 29 cases in the U.S., most of them here in Asia and China, but 10% case fatality. The reason why we stopped this is because when you actually become symptomatic, you're usually severely ill. And that's the peak of virus shedding. You're usually in a hospital, and we learned how to control infections in hospitals. It's not a community-acquired infection like SARS-2. So where did it come from? So human sera collected before the outbreak had no antibodies to SARS-CoV, so this is a brand new virus. The earliest Guangdong cases were in handlers of animals for the exotic food market. These outdoor markets where they bring in food from the countryside, you can go buy it, exotic animals like bats and civets. And these traders had higher prevalence of antibodies than control groups. So it suggested that somehow the virus was there first. The origin, in the following years, they found in a single cave, actually, in Yunnan province, a highly diverse population of SARS-related coronaviruses and horseshoe bats, this one here, Rhinolophus. And in one cave in Yunnan, they found all the elements needed to form SARS-CoV by recombination. Not the exact progenitor, but all the sequences that make up SARS-CoV-1. Now, a key point here is that there were no SARS cases in Yunnan during the outbreak. So Yunnan is significant because that's where they found the bats harboring these SARS-like coronaviruses. And they never identified a direct progenitor. They just identified all the sequences in different bats in that cave. So what they thought happened is that the progenitor emerged in bats, perhaps in that cave, and then infected civet cats. Here's a civet cat. These are being sold in the live meat markets. And then the cats are transported to the markets. So that's why. So no humans got infected in the countryside. It was the civets that were infected. They're brought to market, and then the civets infect humans. So we go bats to civets to humans. We don't know where the bats infected civets, but the civets are raised outside, and so it's possible they were contaminated by bats. But as I'll show you later, we have viruses in civets that are very, very similar to SARS-1. Now, that died. SARS-1 died after a year, and we said people stopped paying attention to coronaviruses, and then in 2012, a new coronavirus emerges, the index case, a 60-year-old male parent who died of pneumonia and renal failure, and they isolate MERS coronavirus from that patient and find that it binds dipeptidylpeptidase 4, which is different from SARS-1, which binds ACE2. And again, closely related to bat coronaviruses, but this is not SARS coronavirus. This is a different coronavirus. This one is spread from camels. It's in fact endemic in camels in the Middle East and Africa. Remember, those camels that got there from the western U.S. many years ago, now they have a MERS coronavirus infecting them. They probably originally acquired it from bats, but many, many years ago. And so this is the source of human infections. These camels are highly prized in the Arabian Peninsula. They're used for labor, they're used to eat, they're used for racing, they're used as pets. So there's a lot of human-camel contact. However, there are some infections that don't have a camel source. We don't know where they're starting. How is it transmitted to humans? Probably via camel secretions. Every camel, when they're born, they get infected in the camel barn, and most of them recover. They get a respiratory infection and they recover, and so probably that's the source of human infections. But it doesn't transmit well among humans. It does not establish chains of human transmission. Every case or two is a new spillover event from a camel, essentially, very much like Ebolavirus. We're worried that it might spread eventually, but this has been going on for a while. There's a case or two a month. It doesn't seem to acquire transmission potential among humans. And again, a camel vaccine has been developed, another one world health approach. This is a rare infection. You don't want to immunize all humans, so you immunize the camels instead, and that will protect the humans. Here's a summary of all the cases. 2,468 cases up to the present since it was first discovered, 851 deaths. CFR is quite high, 35%. So most of the cases are camel to human transmission in the Arabian Peninsula, but sometimes infected people travel, and there have been travel-related cases in the U.S., in Europe, Africa, Morocco, I believe. There was even a hospital outbreak in South Korea. A patient came and someone traveled from Saudi Arabia, got sick in South Korea, was admitted. They didn't know what was wrong with him, and by the time they figured it out, he had infected a lot of healthcare workers, right? So they were able to contain that. So these are all exported cases. You can see the epidemic curve, sporadic cases continuously. Here's the MERS in the Saudi Arabia in 2019, right? January 14, February 57, March 22, etc. Kind of going down here, down to 3 a month. As you raise awareness for the danger, you be careful. Don't kiss the camel. And that brings us to SARS-CoV-2, our third pandemic of the 21st century. This begins in December 2019 in Wuhan with a cluster of pneumonia cases of unknown cause. Because of SARS-1, where we were quite sure that the origin was the outdoor meat market, in the same situation, the Huanan seafood market, where they also sell meats, was suspected as an origin because a lot of cases were associated with that market. However, when the first outbreak was published, it became clear that the very first case, which was December 1st, had no seafood market exposure, as did not many of the other cases as well. So, being a market, it attracts a lot of people, and infected people probably went there, but that probably wasn't the origin. And, where it began, we really don't know. Of course, as you know, since it made its way out of China, it's infected 130 million people globally with 2.8 million deaths. R naught of 2 to 3, and 80% of infections are either mild or asymptomatic, very different from SARS-1. So this is community spread, much harder to contain. When we do the sequence of the genomes of SARS-1 and SARS-CoV-2 and bat viruses, this is what we find. So up at the top is the SARS-CoV related lineage, the original SARS. So here at the very top in shaded blue, these are human and civet isolates. You can see the degree of genome identity is very high. So that's why we think the civet virus is the ancestor of the human virus. And then in bats, there are highly related SARS-CoV-1-like viruses circulating in bats. And these have been isolated many times. And as I said, in one cave, you can find isolates that together make up the genome of the human isolate. Now, SARS-1 is quite different from SARS-CoV-2. This is the SARS-CoV-2 lineage here in red. And these are all SARS-CoV-2 human isolates from different countries down here. And above them are the closest known viruses from bats and pangolins. So this bat coronavirus, RATG13, is the closest isolate that we have to SARS-CoV-2. It has 96% nucleotide identity. The other bat and pangolin isolates are less identical, but they're still quite close. However, these are not ancestors of SARS-CoV-2. They're too distant. This 96% means still 1,200 bases distant. So it shares a common ancestor, but it's not the ancestor as the civet is likely the ancestor of the human virus. So we have yet to find the ancestor. We might find it if we look for it. It's not clear where it would have arisen because you can find similar viruses in Japan and in Thailand. So it may not have arisen in China. Who knows? We have a broad range to do wildlife sampling. But it's quite clear that these viruses emerged from nature. And, you hear all the time people accusing, even the former CDC head. I don't know what got into him. He thinks it came from a lab. It's just not consistent with what we see here. Nobody had any of these viruses in a lab. And more importantly, the recent WHO report failed to find any cases of pneumonia associated with the lab in Wuhan, aside from the ones that have been reported. And they combed through many, many, many cases and samples, everything negative. So what the ancestor is remains to be seen. I think we need to find it, so perhaps we can stop this in the future. By the way, these pangolins were harvested a number of years ago. They're illegally traded animals. I think some of them were harvested in Malaysia and others here in China, as you can see. But their origin is not clear. We don't know where these came from, but they're a number of years old, so they predate the human virus. So they're not the source either. Nevertheless, this is a zoonotic infection, and this is a virus that's become a human virus. It is now transmitting very well among people. It's quite clear that there has been a recombination event that has led to the emergence of SARS-CoV-2, and I want to share that with you here. And so there's the spike gene, right, which the spike itself, which we've talked about a lot. Here's the whole genome of SARS-CoV-2. And if you do a phylogenetic analysis of the whole sequence, you get this tree at the top. And as I've told you, the first isolate, the Wuhan isolate of SARS-CoV-2, is very close to bat RATG13 in this phylogenetic tree. But if you just take the receptor binding domain sequence and do the phylogenetic tree, then you get a different picture here. You get the RATG and the Wuhan isolate clustering with SARS, the original SARS, whereas in this tree it's quite distant. What this means essentially is that the spike probably arrived in SARS-CoV-2 by a recombination event. And so basically what we think happened is that there was a recombination event in the lineage leading to SARS-1 as well as in the lineage leading to SARS-CoV-2. This probably happened about 10 years ago, 92 to 2009. And then the virus diverged. On one branch, it became RATG13, and on the other, SARS-CoV-2. You know, this kind of thing happens in nature all the time. Coronaviruses are highly recombinogenic because the way their mRNAs are made facilitates recombination, movement of polymerase on the genome. And so in their hosts, bats and what other animals they are hosts in, recombination happens a lot. And that combined with mutation provides pools of viruses. All you need to do is encounter them. And again, how this one happened, we don't know, but I've already told you the story of SARS-1. So this is the way the coronaviruses have emerged. We have 4 common cold-causing human coronaviruses, right? NL63, 2290, OC43, and HKU1. These circulate extensively. They infect most people on the planet. They caused very mild upper tract infections. These all originated in some other animal, 2 of them in bats, 2 of them in rodents, in 2 cases via an intermediate. When this happened is probably a long time ago, many, many years ago, and they have since become human viruses. But they were originally zoonotic events. And then we have the 3 epidemic human coronaviruses, SARS-1, MERS, and SARS-CoV-2. And I've told you SARS-1 went from a bat to a civet to humans. MERS goes from a bat to camels many years ago, and more recently camels to human. I think it's highly likely that the ancestor of SARS-CoV-2 came from a bat. Whether there's an intermediate or not, we don't know. I don't think it's necessary, as you can see, for some of these other coronaviruses. I just want to point out that there's a disease of pigs, severe acute diarrhea syndrome coronavirus, SADS coronavirus, which just was detected a number of years ago on pig farms in China. And that's a virus introduced to pigs from bats. It's a spillover event, and then it spread from pig to pig. So these spillovers do not just affect people. When you grow large numbers of animals like pigs and chickens, you will get spillover events into them as well. But you see, it's consistent with the emergence of all the other coronaviruses. There's a reservoir in nature, and it spills over. Not in a lab, folks. Came from nature. I highly recommend you go back and listen to this TWIV, which I did. I visited Australia, where they worked on the Hendra vaccine, actually. And Linfa Wang did that work. He's also working on SARS-CoV-2. He worked on the original SARS. He's now in Singapore, but he's the real Batman. He does work on bats and bat viruses and tries to figure out why bats have so many viruses in them. To end it up, I just want to point out that many of these viruses are really dangerous, Ebolavirus and Nipah and Hendra, and you need to work at biosafety level 4. Now, SARS-CoV-2, you work at biosafety level 3, but these viruses for 4 are very high mortality, person-to-person transmission, and no vaccine or antiviral is available. And so in a BSL-4, you have to wear a spacesuit. Well, it's not really a spacesuit, but it's a specially designed suit, which is sealed, and you have air pumped in, as you can see here. And there's a whole procedure for working there. I actually made a documentary at the one in Boston before it opened. Otherwise, you're not allowed to go in. And you can see that one here. It's quite interesting. That's never been done before. Nobody's ever made a documentary in a BSL-4, so they let us in. They let TWIV in, and you can see us learning how to get suited up and so forth. For example, these suits, they're like $4,000 each. Before you put it on, you have to fill it with air and make sure there are no leaks. And they do develop leaks, and you can repair them up to a certain point. But this one that I used here in Boston, this is very heavy, so if you do animal experiments, they won't claw through it. So how common are these host range jumps? I think they happen a lot. I think people are always acquiring viruses from animal sources, but they don't go anywhere. They're dead ends, or they enter you and they never even reproduce. The ones that we notice that make sustaining transmissions are really rare, and you can count them by the pandemics and the spillovers that we've talked about today. I don't think we can predict them. First of all, we don't know all the viruses out there. We have barely any sense of what viruses are in North American bats. There's hardly any surveillance done. People don't want to support that kind of work. I think it's really essential to do it, but it's hard to get it supported. And of course, it's hard to get people to do it. You have to go off for a couple of months and do wildlife surveillance, and not everybody wants to do that. But I think we need to know what is out there in certain kinds of animals, and we've talked about them today, and be prepared. We knew after SARS-1 that there were SARS-1-like viruses, coronaviruses in bats, and we found out that they could actually grow in human cells in the lab. So we could have been ready with antivirals, but we weren't, and that's the tragedy of this pandemic, that we actually had all the information that we needed to stop this pandemic. We had an antiviral that would have worked. Molnupiravir, we talked about a couple of lectures ago. That was developed many years ago, and it might be useful in this outbreak. It could have been ready at the beginning and stopped this out of China or wherever it began. The bottom line here is that we are nowhere near ready enough for the next pandemic, and there's going to be one. There's going to be a flu pandemic. There is going to be another coronavirus pandemic, probably in 10 years or so. And I hope this experience makes us ready. I'm not sure. Next time, we're going to talk about the biggest pandemic of modern times, which is a zoonosis originally, HIV and AIDS.